Thanks to Michael and to Richard and Danielle and the other organizers for having the chance to talk today. It's great to see so many familiar, well, maybe not faces, but so many friends on the call. I am, for those of you who don't know me, currently working at the Bill and Melinda Gates Foundation. I've been there for the past couple, 2.5 years or so. And before that time, I was at UCSF, starting as an intern in 1982 at San Francisco General, and have been working on HIV and AIDS since that time. One of my last stints there was to work in a collaboratory, the Delaney Collaboratory that I ran with Steve Deeks and Sharon Lewin. And I was interested after all that time in understanding whether there might be a way to bring curative interventions for HIV to all people, because it seemed to me that the NIH was most interested in what was happening in the States, and the biotech companies, as Bora suggested in his previous talk, can only make an economic model to bring any kind of therapy to people that can pay. And I cast a net, and I was convinced by the leadership at the Gates Foundation that they would be interested in hearing the development of a program that could do that, to bring safe, effective, accessible curative interventions for HIV to all. And by their so-called territories are low and middle income countries, the poorest of the so-called Gavi countries. But I think this is, my talk is also, and the program that we've developed is also meant to bring curative interventions to all in the United States. As you know, there's a tremendous inequity with respect to delivery of healthcare in all parts of the world. The rationale, let's see, and I'll solve it the way that Michael Adams, there we go. The rationale for the program, I want to put in a slightly different way than what's been discussed in the past. Half of the people living with HIV in the world are not able to get or to remain on antiretroviral therapy. Most of those people live in places where much money has been poured over the past couple decades, but that money is dwindling. There's less and less support. And of course, those places include the East Palo Altos and other spots in the United States that don't have appropriate infrastructure. And that means that transmission will continue, especially in Africa, where there's a so-called youth bulge. There are more babies being born because we've actually done something there. And those babies are, in a heterosexually oriented epidemic, more women than men actually in Africa are infected in childbearing age. Those babies are being born in many cases with HIV. So what the challenge is to figure out how to actually bring cures of this type that are durable and safe and accessible everywhere in a way that can be accepted by people that either do not have access to medical care or do not want to go actually into medical care for a variety of reasons. I learned about, when I went to the Gates Foundation, something called a target product profile. And many of you have probably heard about this, but that's what drug developers, of which I am not, think about when they think about what they want to do down the road. Where does this drug, what it's going to look like, who's going to take it, and how they can get it out. And to me, in a very simple-minded way, it seemed that the best profile would be a single shot, in and out, done and gone. Intravenous, as Chris mentioned, percutaneous intramuscular would be even better. I stole this from Chris's review that he showed from 2019. That shot would, well, I'll describe it, Chris has already intimated to you, that could contain a vector that carries a cargo to long-lived cells and confer upon those cells the ability to resist HIV. If so, then, oh shoot. One thing that would have to be clear is that it reduces the viral load to the point where the individual who gets it doesn't get sick and also is no longer able to transmit to individuals with whom they have contact. Other really important attributes are listed here, one of which I think the third there bullet point is really important. It has to be something that not just reduces the viral load, but it also has to prevent reinfection upon exposure. So Timothy was on PrEP, right? Because he, although cured, did not want to become reinfected. That's not a suitable way for an accessible cure down the road. So we need to have thought about how that can happen. Importantly, what this means is that it's likely not necessary to eradicate all of the reservoirs of HIV that are present, but to reduce them and to then keep them at bay, likely with an immunologic effect that is hopefully similar to that which might be found in so-called long-term non-progressors or elite controllers. I saw the question on the chat. How do we do this? So the way the work is divided out, and I'm happy to send at any time papers to people or to field phone calls or emails to go through the detail of all of this, but in very short form, and I'll go through these points in a little bit more detail, there's really 4 buckets of work, okay? And this is what we're doing now. You have to have the right design. What is the intervention that is going to be put into the cell that allows that cell or its progeny to suppress HIV? Chris mentioned some, Kim and Boro also. Those are designs that could be there for HIV, although I must say that I think we're still in the infancy of understanding what the right design is for HIV. Secondly, once it's introduced, it has to be durable. Hence, the introduction has to be in a cell that lives for a long time or that does something called self-renew. That's a characteristic of a very small number of important cells in your body, the hematopoietic stem cell that Chris showed you being one of them. T stem central memory cells do that, B cells do that. So those are 3 good targets for durability, and we have to figure out ways to target those cells in a safe manner. Thirdly, we have to be able to detect the virus that might come back, because for sure, in resource-limited parts of the world and elsewhere, the initial in vivo and ex vivo therapies are going to fail. And not all people have access to come back to the clinic every week. So there has to be basically a home test that allows individuals to understand if a rebound is occurring. Ideally, it would be a non-viral circulating biomarker, which is put into a home test. Not less importantly, such biomarkers would allow us to understand if we had something that reduced the size of the reservoir. Delivery is a really important issue. I think exemplified by the slow uptake of PrEP. It has been there, right, for almost 10 years, and it's still not being taken up. So we have to prepare the world, and PrEP is nothing compared to gene therapy. If we're going to bring gene therapies into the world, we have to start talking about that now with the key stakeholders and the community. One of the reasons I'm here today, so that there is knowledge and socialization and feedback. That's how this epidemic has worked from the get-go, and I think that's how it has to continue on if we're going to reach a cure that's accessible. These 4 things, the leadership at the foundation thought were worthwhile funding. It's clearly a very ambitious program. It's not going to happen overnight. We, as I mentioned, don't have the right, I don't think, design for curing HIV just yet by this manner. We do have actually a great deal of information about how to cure sickle cell disease, okay? So I want to put that out there. If you can believe it, the Gates Foundation didn't actually have on strategy cures for sickle, even though there's many, many people in sub-Saharan Africa that are born with it and that die by the age of 5 and have horrible lives if they continue further. But we know we just have to correct one small change in the hemoglobin chain in order to cure people of sickle cell disease. So we've posited that sickle cell disease could be a way in which we learn how to actually target hematopoietic stem cells in vivo and make the changes, how we can deliver those into resource-limited parts of the world. And leadership also bought into that and said basically, okay, go forth, but create partnerships, because there's no way with a goal such as this that's going to take so long that it's either going to be done by a single academic lab or even a collaboratory of academic labs or a biotech company. It's going to take a partnership of the type that Boro is building. And this is exactly right. Unless we have those partnerships, it's not going to go forward. So the program as presented, developed over the year of 2018, was funded $100 million over 4 years in early 2019. we immediately began talking to the NIH and within months set up a 50-50 collaboration with them to do studies on in vivo gene therapy for sickle and HIV. They were to have put in 100 million, but they've since that time actually put in far more for studies of this type to catalyze the field. It's highly risky work. And it's not the kind of thing that most graduate students or postdocs see as a thing that can move forward, but it's with incentives, it will. The work that we're doing in this collaboration is not meant to detract from work that is ongoing for what might be called standard of care for sickle and HIV. Those, the red and the blue circles on the opposite sides, but more on the center. It's focused on shared gene-based strategies that could be used to treat each disease. And that relates to, Chris talked about vectors. I'll talk about them a little bit more too, to understand which ones tropism means, where do they go? And do they go to the right place and not to the wrong place? Do they get there efficiently? Do they modify the cell well? If they modify the cell, how do they target it? You've probably heard of CRISPR. There's many other ways now in which modifications can be made, and we need to figure out which ones work best. And not least, we need to figure out how to get these things packaged together so they can be given in vivo. And what's been happening over the past several years, is that this corpus of money has been
doing for homework today.
I think somebody, well,
My what?
you might be.
A health thing.
Somebody's homework is being done.
Scientific studies to cure diseases.
Anyway, so these funds are now being put out in the way of grants, mostly to academic labs from the NIH. From the foundation, initially, it was very difficult to fund, believe it or not, well, you probably would believe it, academic labs or biotech pharma, because the foundation requires, as a philanthropic organization, that any funded development be there for the foundation to use in resource-limited parts of the world. And that infringes upon property rights, so that it was difficult, but we finally, I think, cracked that nut. And the so-called humanitarian license has now been accepted by all of the universities in the United States and Australia and Europe. And this last year, 2020, we were also able to invest in a total of 6 small to mid-cap biotech companies, and also into Caring Cross, the organization, nonprofit that Boro just discussed, which moves us even further, right, from the basic research place of the academic lab to the clinic, because biotech pharma, especially the small and mid-cap companies, can do that. We've also been able, from the foundation, to make what are called charitable equity investments, where we've invested money in companies. You may have heard of Immunocore, they're in England. I'm sure you've heard of BioNTech. And with these equity investments, we can actually push them to do work that allows their talents, and they have considerable talents, to be moved into resource-limited parts of the world. And Immunocore is making something that I think will be useful for reduction of the reservoir size, and BioNTech is making something to target B cells, actually, for modification of them. They're still relatively small to mid-cap biotech companies, and just recently, last fall, we announced it's been a 2-year program to stand up a dedicated workforce in the hallways of Novartis. It's called the Institutes of Biomedical Research. It's 25 people that are going to be focused on delivery in vivo gene therapies for sickle cell disease and HIV. And this is really important because it means that it is aspirational, and we have a long way to go. But we have a multinational company with chops now that knows how to bring things into the field working on this. And they're doing this in a manner which I find to be very charming, in the sense that I do believe that upper management at Novartis really does want to get these kinds of therapies to people who are not able to afford other kinds of therapies, and I believe they'll do that. We're also working with a couple companies now to see if we can help them. They can help us to work on this biomarker issue. We haven't successfully landed that yet. The first task at hand, amongst all the different things that we need to do, is can we target and edit an hematopoietic stem cell in vivo. There's lots of challenges. Chris went through a bunch of them. We now have, I think, probably a total of 20 to 25 different viral and non-viral approaches that are being funded in different labs in academia and in biopharma. And we're going to do a bake-off, okay? We're going to understand, of all those that are there, which ones look good. We'll fund those. We've worked with Chris's colleague, Stefan Radke in Hans-Peter Kim's lab, to set up a humanized mouse core at the Hutch so that we can put them all through the same model and do head-to-head comparisons. We're actually thinking we might get to this done by the end of this year. And the comparisons, we're going to ask to show us which of the vectors actually have the best biological activity, that is, which ones get to the stem cell and not to the other cells that we don't want. And of those, and we already know that there are some, one of them from Genadair has that gold nanoparticle that Chris showed you is beautiful. And the adenoviral vector that Hans Peter and Andre Lieber have been working on is also great. And there are others, okay? So we're gonna ask the question of those that are biologically active, which ones are likely developable? That is to say, can be made at scale with low cost of goods so that we don't have to charge people $2 million, right, for a therapy. And those that look good, of which I think there will be more than one, will go into a non-human primate core, which has been set up at the University of Washington Non-Human Primate Center, again, run by Hans Peter, which has been made GLP-like, meaning to say it's got more precision to it with respect to the procedures and practices than most cores would have. So we think that it will be IND enabling. That is a straighter shot to the clinic. I'll spend a couple minutes on the last two Ds, okay, and then I'll stop. With respect to detection, it's a problem. We know, right, that there are lots of genomes in individuals that are on suppressive therapy, 10 to the 9th to 10 to the 10th. Of those, only about 0.1% is full length and likely to be replication competent or rebound competent. But it's even more difficult than that. Of those that are full length, there are only a few that live in so-called genic regions as opposed to gene deserts. Qualitatively, these full-length genomes are more likely to rebound than these. And this work is all very recent, right? We're just beginning to understand what's going on with the reservoir. The companies haven't wanted to touch HIV Cure because there is no biomarker. They don't want to spend the money to work on therapies without a biomarker. Academic labs, this is highly risky, once again, work to do, so it's hard to motivate academic labs to do it. So what we've done out of the foundation is to set up what we call the Reservoirs Consortium. It's a 5-team, 40-lab, 3-continent exercise of collaboration over a period of 4 years to discover circulating non-viral biomarkers to assess the quality and quantity of the rebound competent reservoir, and importantly, to know when curative interventions that come down the pike fail so that people can go back on ART. And we hope by early 2022 that we'll have such biomarkers identified. It's likely going to be a constellation, more than one, and then work with a multinational company to develop it into a kit that can be used for diagnostics. Finally, distribution, big problem, right? To bring things into anywhere that's new, let alone gene therapy in sub-Saharan Africa. We held a meeting early 2019 in which we convened many people from really all over the world, but especially low and middle income countries, to ask the question, if we ever do have a therapy that might be a cure, how are we going to bring it to you? And from this meeting, were organized 2 working groups that just published late last year papers that on the one hand talked about target product profiles. This is similar to the slide that Boros showed. It's going to be an evolutionary progression from very complicated combination therapies through to ex vivo types of gene therapies, to in vivo gene therapies, to a shot in the arm. And that all has to be done in parallel with cross-checking back and forth for efficacy and safety. And then we've also set up a public-private partnership called HCAP, for HIV Cure Africa Acceleration Partnership, to bring together key stakeholders in sub-Saharan Africa to ask the question, how if there is a curative intervention, should it be introduced? What are the, how do we engage the community? What is the economic model? How do the regulators see it, et cetera? And that's being run by Mark Dybel, who's, I think many of you know, was the PEPFAR ambassador for many years. And we are working with many people from sub-Saharan Africa in this group now too. So I'll stop there, and I want to stress that this is very much upstream research. It's not going to come to your clinic soon. It will be data driven, and it will be moving forward if the data look good, and it will not move forward if the data show lack of safety or lack of the ability to make it accessible. So I'll stop there, and I'm happy to, like I said, answer questions now or anytime if you want to call.
Great.
Thanks.
Thank you, Mike, for that. And thank you to all our presenters. This has been a fabulous download of lots of information that we have. And we did have one little last part that we wanted to get through before we get to our questions. And so, Gary, I saw that you've signed on. If you wanted to unmute yourself. And Adam, are you on the call? If so, please unmute yourself and speak up so we can hear if you're here.